Hello, my name is Saul Laredo, and this is going to be part 2 of the first lab for cell biology. And in this video, I will explain to you my concept map. So, here is my concept map. I'll zoom out a little bit so you can see the whole map. It goes from here and finishes over there. The resolution on this video may not be really good, but I will zoom in whenever I explain these, and I'll make sure that you guys will be able to read this, okay? I'll begin with the atom, the most single basic unit of chemistry. The atom contains a nucleus, electrons, protons, and neutrons. From the atom, I branched out to the elements. And right here I have the elements that were provided in the instructions. I have them right here, but we will get back to this in a second. Every element has an atomic number and an atomic weight. The atomic number is essentially the protons, and the protons are located inside the nucleus, and that is how I brought this back here to the nucleus. The nucleus is surrounded by electron shells, which are obviously electrons. So that's why I linked it back to the electrons. Electrons give covalent bonds. They are either shared unequally in a polar covalent bond, two atoms are shared in a nonpolar covalent bond, two pairs of atoms will be shared in the double covalent bond, and three pairs will be shared with the covalent bond three pairs of atoms will be shared in a triple covalent bond. Valence electrons are used in the formation of chemical bonds. There are substances that do like what they are called electron donor and an electron acceptor. So there are substances that donate, and there are some that accept, and that is their purpose. One donates, one accepts. We also have ionic bonds. Let me just see right here, ionic bonds. An ionic bond is a bond between an anion and a cation. Anion being a negative charged ion, and cation being a positive charged ion. We also have hydrogen ion and hydronium ion right here as well with the ionic bonds. We also have an interesting one, which is kind of like a bond. It has to do with bonds, but it is not a bond. I'm going to zoom in right here. This bond cannot be... I'm trying to read my handwriting to the screen. Let me just read this. Bond cannot happen because of water. This is the hydrophobic interaction. Hence the word phobic, phobia. So when this happens, the molecules cannot be mixed with water. And here we have water, which is the most fundamental form of liquid, and it is essential to every human being and every living animal. We all need water to live. We have water isotopes, and from isotopes we also have radioactive isotopes. We have here a half-life, which is the time taken to decay, which is a half-life. It records like the time taken to decay of a radioactive isotope. There's also cohesion and adhesion. Cohesion are when the same particles stick together, and adhesion is when different particles, like they're different particles, but they stick together. So cohesion, all the particles that are bonded together are the same, and in adhesion, the particles that are bonded are different. Now, I will let you guys look at this right here that I have of bonds. We also have right here oxidation reduction, and let me just see my notes right here that I have written down. Oh, and the oxidation reduction is a partial or complete transfer of electrons, and that is with my bonds. To the elements we go. Here we have oxygen, hydrogen, nitrogen, sulfur, sodium, chloride, phosphorus, potassium, calcium, iron, and last but not least, carbon. Carbon is very important for chemistry. There is a whole branch of chemistry just dedicated to it, and that is organic chemistry. But let me talk here about the mole first, Avogadro's law, and molarity. A mole is the same number of particles as there are in the atoms of 12 grams of carbon. And molarity is it deals with the moles of solute per liter of solution. We also have Avogadro's number, which is the number of atoms in 12 grams of carbon-12. But as I said before, the most important out of all of these is organic chemistry. Now right here in organic chemistry, and I apologize for this tape. I accidentally wrote the wrong arrow. It was supposed to be this one. I'll get to that right now. First, I'm going to show you guys the functional groups with carbon. Now, in the instructions, we were given functional groups, and we were told to label them and describe them out. And these functional groups of carbonyl, and I apologize if I can't say these correct, ketone, aldehyde, carboxyl, carboxylic acid, and methyl, these all contain carbon. Now here are the rest of the functional groups that we have. We have hydroxyl, amino, amine, alcohol, sulfhydryl, and phosphate. These do not contain carbon, and that is why I put these with inorganic chemistry. Now I'm sure there's a bunch of things right here that have to do with inorganic chemistry, but for the sake of this chart, I just linked the inorganic chemistry with these just because they didn't have carbon in them. Now here we go to the organic compounds. We have carbohydrates, proteins, nucleic acids, and lipids. Our carbohydrates would be our sugars. When it's one saccharide, it would be monosaccharide. When it's many, it'd be polysaccharide. And that is essentially what we have with our carbohydrates. We have proteins, which are made up of amino acids, and we also have right here glycoproteins. These are attached by a polypeptide chain. We have nucleic acids, and these are essentially what carry our DNA. This is what carries our genetic material. It's our DNA, our RNA, all that good stuff. I'm just trying to see. I wrote down right here the difference between the nucleoside and nucleotide. Well, they're both essentially the same. They both consist of a nucleobase. I'm sorry about that. Of a nucleobase and a 5-carbon sugar. The only difference is that the nucleoside does not contain a phosphate group. I wrote right here, essentially the same, but no phosphate group with the nucleoside. This does have a phosphate group. And last but not least, we have our lipids, which are our fats. We have our phospholipids, our fatty acids, glycerol, and our fats. And we have our saturated and unsaturated. Unsaturated being liquid at room temperature, and saturated being solid at our room temperature. And then we have right here glycolipids, which is very interesting. These contain carbs as well. So I was kind of debating on whether to put them in carbohydrates or lipids, and I just put them under lipids because it said lipids in the name. But I also attached an arrow right here to carbohydrates. And this is where I messed up. I'm not sure if you see the beginning of an arrow right here, but I kind of did the same thing and I ended right here. That was my fault. This is my mistake. The last thing I'm going to explain to y'all are the measurements. Now, for chemistry and for well, usually any labs that we do now, physics and all those science classes, well, there's a system we have to follow for our measurements. We have our grams, liter, meter, milliliter, and then we also have a pH scale, which it measures how acidic or how base something can be, or it can also be neutral, which is most of the time that's water. We have salt here, that is sodium chloride, the result of the reaction of an acid and a base. We also have molecular weight. I have right here what I have written for molecular weight. The ratio of the average mass of one molecule of an element to 1/12 of the mass of an atom, carbon-12. And the unit of a molecular weight for protons equals a Dalton. And last but not least, we have electronegativity. And that is a measure of electron attracting ability of atoms in a chemical bond.